Us in this block of contributed talks is Shouvik Mani. Shouvik is a master's in computer science at the Azizia Lab at Columbia University and a research intern in the Pear Lab at Memorial Sloan Kettering Center. His research focuses on developing novel machine learning methods to model temporal and spatial dynamics in single-cell data. And additionally, he's also very passionate about teaching computer science and STEM outreach. All right, Shouvik, take it away.
Awesome. Thanks, Stephen. And hi, everyone. My name is Shouvik Mani. And here I'm presenting joint work with Duran Habib, Russell Coons, and Dr. Donna Pear at Memorial Sloan Kettering. Today, I want to show you a new way to look at your spatial transcriptomic data. As a field for too long, we've been thinking about cells in isolation. And this has come at the cost of missing out on a lot of the cellular microenvironments which also exist in this data. So today, to address this gap, we are introducing SPOT, which stands for Spatial Optimal Transport. And SPOT is a new framework to characterize microenvironments in these spatial data sets. So the question is, why should we think in terms of environments? Well, it turns out that biology is inherently spatial, and from environmental signaling to cell-cell interactions, there are just so many ways in which the spatial context of cells influences cell state, cell fate, and just ultimately orchestrates global tissue function. And we see this play out in cancer, where the spatial architecture of these tumor immune microenvironments, TMEs, play a key role in determining response to immunotherapies such as immune checkpoint inhibitors. So for example, here I'm showing two classes of tumor microenvironments. On the left, we have what's known as immunologically cold tumor environments. And here you see that the CD8 positive T cells kind of lie along the periphery of the tumor. They're actually in physical contact with these tumor-associated macrophages, which prevent these T cells from infiltrating into the tumor core. So this class of environments has very poor prognosis and poor response to this ICI therapy. In contrast, you have these hot tumors where you see that these T cells have actually infiltrated into the tumor core, and they're expressing these T cell activation markers. They're also expressing this PD-1 gene as well. So they're primed to be targeted by these checkpoint blockade therapies. So it's clear that the kind of spatial topology of these microenvironments play a key role in determining response to therapy. And unfortunately, though, our understanding of these environments is rather crude, and it's because the computational methods we have to analyze these environments focus on more discrete analysis, looking at distributions of cell types across tissue. So what we wanted to do is we wanted something more precise, more granular, right? Beyond just cellular compositions, can we actually get at the gene expressions in these environments, and then can we understand how these gene expressions are spatially distributed across these environments? And if we could do that, we could finally leverage these spatial data sets and make full use of them, and understand these kind of spatial mechanisms in both normal biology and disease. So that's what we're going to do today with SPOT. So what SPOT is a framework to analyze microenvironments in spatial transcriptomic data sets. And this framework comes complete with a suite of methods to represent your environments, to compare environments and calculate distances between those environments, and then using those distances to cluster and visualize those environments so that we can understand and we can reason about cells, not based on just their phenotype, but their spatial neighborhoods. So next, I'm going to take you through each one of these methods and show you what it looks like when applied to real data. The first challenge we faced was kind of figuring out how to even represent an environment, right? We know how to represent a cell. We have gene expressions, but okay, what if you have an environment of cells? So, the approach we took is the most simplest and objective approach, where, given a tissue section, so here we're looking at a section of a mouse primary motor cortex, assayed using a MERFISH, which is spatial technology. And each point here represents a cell, and they're colored by their cell types. So as you can see, this nicely reflects the different cortical layers in this tissue. And what we did, so what SPOT does is, for each cell in this section, it looks at the nearest neighboring cells. So here we look at the 30 nearest neighbors to this star cell over here. And using those nearest neighbors, SPOT assembles this cell by gene matrix, which we call an environment matrix. So it's just all of the gene expressions of the neighboring cells just consolidated into one matrix. And this is, just a very simple, elegant representation of a cell's spatial neighborhoods. And once you have this representation, you can finally analyze it, and you can think about modeling these environments, not just individual cells. So, we were very excited about that, and we're excited about how simple and elegant it is. But one challenge we faced was, these environment matrices, they didn't really lend themselves well to being compared using Euclidean distances, which, as you know, is the default distance metric used in basically all of the visualization and clustering algorithms. And the reason why is because just consider this as an example. Let's say you have an environment matrix here, and you just permute the rows in this matrix. So you're basically shuffling the cells in the environment to get a new environment matrix. And notice that, these two environments are identical, right? All we've done is just, shuffle the cells. But if you use Euclidean distance, or in the case of matrices, Frobenius norm, you're not going to get a zero distance, or you're going to get a potentially high distance. And so this will fool a visualization or clustering technique such as UMAP or phenograph, which will think these environments are actually very dissimilar. So we realized that we needed a distance metric which respected the fact that the ordering of cells, right, don't make two environments different from each other. So we needed a distance metric which was invariant to ordering of cells. So what we did is we turned to the literature on optimal transport, which helped us derive such a metric. So here's a kind of cartoon example where we're looking at two environments in the gene expression space. And let's say these two environments are very similar to each other. And you can kind of see that because the cells in these environments, the 3 cells in these environments, have very similar gene expression distributions. But let's say they are the order of the cells in the environment matrix are actually these numbers over here. So 1, 2, and 3, 1, 2, and 3. So you'll notice that if you just use Euclidean distance, then you compare cell 1 to cell 1, cell 2 to cell 2, cell 3 to cell 3. And you realize that's not the optimal alignment, right? You get a high distance. Now, the benefit of using optimal transport is you can consider all of the possible mappings between cells in one environment to cells in another. And you can find the optimal mapping, right? The one that minimizes, in our case, the Frobenius norm. And this is the alignment of cells, right? So we have kind of shuffled the environments in the right way so that we can respect the fact that these two environments in the gene expression space are actually similar to each other. So, once you've aligned them, the distance metric then that we use in SPOT is just the, Euclidean distance after this optimal alignment. And the great thing about this, right, is, first we defined a representation for environments. Now we have a principal distance metric to compare environments. So now you can just do pairwise distances between all of the environments in your data set. And you can plug in these distances into all of the tools that you know and love. And so we did exactly that. So we took, again, this primary motor cortex. For each cell, we defined an environment matrix. We calculated all of the pairwise environment distances, and we just plugged that into phenograph, which is graph-based clustering, to find all of these clusters, which we visualized in UMAP space, which was also calculated using these distances. And then we also colored the cells or environments in situ with these clusters. And notice you see these, beautiful like layers, right? Like, each of these environments, right? Like once you cluster them, they correspond to different layers in the cortex. And, this isn't just by accident, it's because we have a metric which is faithful to how these environments should be compared. So, that's something you could do, right? Use off-the-shelf tools using spot distances. We also went a little further and we built our own clustering algorithm, which is more environment-centric. And what this algorithm does is it uses a style of k-means clustering to spread out environments across the space and learn the canonical or most representative environments, which kind of summarize this data set. So in this case, I'm showing here 3 environments, canonical environments, which we have learned from this data set. And you notice that, these environments nicely map again to these cortical layers. And you notice that some environments like this, bottom left environment over here, these environments have more cellular heterogeneity, more different types of cell types than these outer layers, right? So that's something interesting, which, you couldn't tell from all this noise over here, but you can tell from these clusters. Again, beyond just, cell type, compositions, which, other methods can do, really the benefit of this is, we think of the environment as like a first-class citizen, right? So what you get as a result for each of these canonicals, you get a representative environment matrix, right? So, you can say, what are the typical cells in this environment? And hey, by the way, look, for this specific cell type, they're not all the same, right? We see a gradient of expression in a specific cell type in a specific environment. And I think this just opens it up to a lot of novel biological insight. Finally, we also applied SPOT to embryonic development data, where we applied it to all the environments in the spinal cord, and we visualize these environments using a force-directed layout. Again, using spot distances. We colored them using the first diffusion component using spot distances. And the magical thing, this is truly amazing, is that the first principle or the first diffusion component, right, the axis of the highest variation, just neatly maps to the anterior posterior axis, right? And this is telling you, right, like the environments in this part in the anterior is very different from environments in the posterior. And it's really nice that, SPOT can kind of recapitulate this kind of like known phenomenon, known biology. And next, I'm really excited to release SPOT in the coming weeks and to see what's kind of novel biology that SPOT learns in, for example, tumor microenvironments. So there's definitely a lot of potential. I'm excited to see what the community can come up with. And finally, I should mention that I'm on the job market this year. Specifically, I'm looking for a low-wage job for the next 4 to 6 years, preferably 4. But I mean, if it's 6, I'll work with it. What can you do? So if you like the stuff I'm talking about, check out my website and definitely get in touch. Over the last year and a half, I've been applying machine learning to these various intriguing and compelling problems in biology. And I love the stuff and I want to learn more because this is my idea of a good time. So get in touch and we can chat. I just want to thank all of my co-authors, Duran and Russell, for actually conceptualizing this, and Dr. Pear for her guidance. And so now I just open it up to questions.
Great. Thanks for the wonderful talk, Shouvik.
Thank you.
Excuse me. So Rebecca actually has a question. How does the optimal transport distance handle environments with different cell types or numbers of cells, given that it relies on matching cells?
So the way we've defined the environment matrix is that you have the same number of cells in each matrix, right? But you're right in the sense that different kinds of environments have different types of cells, and so that's a parameter that you can play with, right? You can actually expand the size of your original environment matrix to kind of get at that.
Great. All right. So with that, thank you again to all of our contributed talk speakers for a wonderful session, and I'll be handing over the moderation duties to Eli, who will be introducing our next keynote.
Thank you.